Can somebody confirm you can hear me?
I can hear you.
Thank you. Go there. Before we begin, are there any questions? All right, let's begin. So today we're going to be doing Lab 9. Today is August 12th, and do realize that you have a quiz 4, tomorrow on Wednesday. I'll try and get quiz 3 graded tonight. And I also want to let you know the infectious disease paper is due 11:59 p.m. this Saturday. If you've not okayed a topic by me, you need to get the topic okayed by me. There are some topics that I divide them up, and there are some topics that, I don't know. You just need to get it by me. Only one person can have one topic, so there should not be anyone doing a topic someone else is doing. Any question about any of that? Boy, we're getting towards the end of the class here. All right, today we're doing Lab 9, identification of gram-negative organisms. This will be our last real lab. We still have the infectious disease paper to turn in this Saturday, and you still have the unknown project to work on. But today will be our last lab. And you do have the unknown notes pages, which will close on August 26th. And then the unknown report is due Wednesday, August 27th. Any questions about any of that? So actually, we only have 2 more weeks of the term left. All right. Lab 9, ID or identification of gram-negative bacilli. You need to complete Lab 9, complete the case studies, and complete the Lab 9 worksheet. There is some reading for this lab, but not very much. You should read page 131 and 132 in chapter 5, biochemical tests and bacterial identification, and then chapter 10, methods of classifying and identifying microorganisms, only 4 pages, page 272 to 276. In today's lab, there will be some video clips. The learning objectives. Upon successful completion of this lab, students should be able to interpret the results of the following biochemical tests. The Imvik reactions. Imvik is an acronym for the indole test, I, indole, M, methyl red test, VP, the V for Voges Prestere or VP test, and then the C for citrate. So the Imvik is actually 1, 2, 3, 4 different tests. You should be able to understand and give the results for the carbohydrate fermentation test. And that could be many different sugars. I won't ever ask a question where you're supposed to do like 5 different sugars, but it could be glucose, sucrose, lactose, mannitol, and inulin. So 6 different sugars. You could have other sugars, but we don't have that in the table. Motility, give me the results of the motility test and the hydrogen sulfide production test. Explain how to interpret TSI, triple sugar iron slants, the SEMS deep, that's a sulfide indole motility test, and then interpret the growth on McConkey's and or MAC and HE or hectone enteric auger plates. Any question about any of that? All right, and then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. You will have case studies on the final, so you need to know how to do that. And this will be a very good practice for doing the case studies.
I'm sorry, can you say that one more time? What is gonna be on the final?
You'll have case studies on the final,
Oh, okay, thank you.
okay? So this lab will be helping you for the final because you're going to have to go and do some case studies. And I know some of you are working together on these because you have the same wrong answers. And I also know that you can easily do the case studies by going online, especially just putting in the information in artificial intelligence and getting the answer. But realize you can't do that on the final. So it really helps you to work on it on your own. Members of the family Enterobacteriaceae are all small gram-negative non-spore forming bacilli. They are all facultative anaerobes. The family Enterobacteriaceae has 53 genera. 24 of these genera are known to cause human infections. Below are some common genera isolated from clinical specimens. All of the members of the family Enterobacteriaceae are all glucose fermenting and oxidase negative organisms. Most of the genera are normal microbiota of the intestinal tract of different animals, aka we call them coliforms because you can find them in the intestines or the colon. But they can be opportunistic pathogens in that they cause urinary tract infections, wound infections, pneumonia, septicemia, and a host of other diseases. Some common genera responsible for causing opportunistic infections include E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Oh, good. The lecture file just finished. My computer will speed up. Several genera of the family are also associated with gastroenteritis. These include organisms of the genera Salmonella, Shigella, and E. coli serovar 015787. Normal E. coli is found in people's guts, and it actually helps us digest our food. It does not cause any infection when it is growing in the gut. Obviously, if it's growing elsewhere, it could cause an infection, but normal E. coli does not cause stomach flu or gastroenteritis. However, E. coli serovar 015787 is a human pathogen that causes gastroenteritis. And I think you all know Salmonella and Shigella are also human pathogens. You don't want them growing in your gut. Salmonella, in particular, is found in the guts of birds, especially chicken, but other birds as well. And if you get it in your gut, you'll get either salmonellosis or typhoid fever. Any question about any of that? Pseudomonas is a family similar to family Enterobacteriaceae in that they are gram-negative bacilli. However, all members of the family Pseudomonaceae are non-glucose fermenting, and they're all oxidase positive organisms, as well as the fact that they are aerobes. So you can differentiate the family Pseudomonaceae from the family Enterobacteriaceae from one of three tests, meaning Enterobacteriaceae, they're all facultative anaerobes. They all can ferment the sugar glucose, and they're all oxidase negative. And then the family Pseudomonaceae are all aerobes. They cannot ferment the sugar glucose, and they're all oxidase positive. Pseudomonas aeruginosa is the most common human pathogen in the group. It is an opportunistic pathogen causing infections, particularly in immunosuppressed individuals, burn patients, and trauma patients. Fortunately, Pseudomonas aeruginosa is not good at causing an infection in a healthy individual. It has a, It's not good at breaching the intact skin or intact mucous membranes. But if those are damaged, it can cause an infection. Pseudomonas aeruginosa is also implicated in healthcare acquired infections, also called nosocomial infections. And it can be found in many places in the hospital. Any place essentially that's wet. It's important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli cultivated from clinical specimens. In this lab, we're going to look at tests, biochemical tests, that will help you identify gram-negative bacilli. And here's a whole bunch of tests. We're not going to look at all of them. And there's one where you're doing all 12 tests at one time. We'll talk about the indole test. Some organisms can produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added to the tube. Kovacs reagent can react with indole to producing a red color at the surface. And that's not very red. I'm not sure why it's so purple there, but it should be cherry red if it's indole positive. If it's indole negative, it'll be yellowish in color. Klebsiella pneumoniae does not produce indole, but Klebsiella oxytocca does produce indole. All right, the next two tests I'm going to talk about are the MEFA-RED and the Voges Percure test. And I'm not Russian, so I could be butchering this name. I'll generally just call it the VP test and the MR test. This begins by inoculating one tube. You then grow the tube, and then you split the tube into two portions. One portion used for the MR test, the other portion used for the VP test. So you inoculate one tube, and then you split it into two, and then you can run two different tests. Any question about any of that? Both the MR test and the VP test are used to look at the end products of glycolysis. If acid products are made, we can determine that from the MR test. But if neutral products are made, we can determine that from the VP test. So initially we have one tube that we call the MRVP broth. It has both peptones and glucose in it. You inoculate it and grow it from 2 to 5 days. You then take the tube out of the incubator and use half the tube for the MR test and then half the tube for the VP test. For the MR test, what you simply do is add phenol red to the tube. If there's acid products in the tube, the MR test will turn red. The methyl red is red, by the way, and it will stay red if it's acid products. However, if it's not acid products, then the MR test will be negative and it will change from red when you first add it to yellow. And that happens almost immediately. Any questions about the MR test? In the VP test, we're looking for a neutral product made from the glycolysis of sugar. And what you do is you take half the tube from the MRVP broth, and after it's grown for 2 to 5 days, you split it into two, and the portion that's used for the VP test, you first add Barrett's reagent A and Barrett's reagent B to it, and then you shake the tube for 1 minute to get oxygen in the tube, and then you let it sit for 60 minutes. If acetone is made, it's a neutral product, and Barrett's reagent will turn red in the presence of acetone. So acetone will turn red if it's VP positive. If it's VP negative, it will remain yellowish in color. Any question about any of that? So an easy way to remember these three tests, the indole test, the MR test, and the VP test, if ever the test is positive, it will be red or reddish in color. If ever it's negative, it'll be yellowish in color. Any question about any of that?
I have a quick question. It says that the broth needs to be incubated for 2 to 5 days, and that's not like if we were using this test for our unknowns project, could we use the broth that we already have
For what?
for this test, or do you have to make a new, because it says containing peptones and glucose?
I still am not sure what you're asking. If you ask for the MR test, I will just take the tube, set it up, and then 5 days later give you the results of the MR test. If you ask for the VP test, I will give that tube, the results to the tube after 2 days.
Okay, and those would be 2 separate tests.
Those are 2 separate tests, yes.
Okay, perfect.
Yeah.
Thank you.
I've never had a student who did one test and then asked for the other test. I suppose if that were to happen and you did the MR test first, I would give you the VP test immediately. But with the VP test, you only need to grow it for 2 days, but the MR test, you really need to grow it for 5 days. And so you wouldn't get it immediately.
Okay, thank you.
All right. Oops, let's see where we are. All right, so let's move on to the last of the IMVIC tests, citrate utilization. This test is used to determine whether bacteria have the ability of using citrate as its only carbon and energy source. Simmons citrate is put into the tube. It's grown on a slant. If the bacteria can utilize citrate, the bacteria will grow and it will change the color of the media to blue as alkaline products are made, meaning it'll up the pH and turn blue. If the bacteria cannot utilize citrate, the bacteria will not grow and the tube will remain greenish in color. Any question about that? I can mention that the pH indicator is bromothymol blue, which at the pH you start it with, I don't remember what that is, but something around the pH 7. It's greenish in color, and if the bacteria can grow and make products in the tube from citrate, it will change the pH and make it more alkaline, and the tube will turn bluish in color. E. coli cannot metabolize citrate, so it will stay green. Let's see if I have an example of one that does. I think Citrobacter turns bluish. Citrate is used in the Krebs cycle. And so the bacteria, if it can use this, can go through the Krebs cycle and then aerobic respiration, and then will grow on the plant. If it can't use citrate, then there's no carbon source, there's no energy source. The bacteria won't grow, and the pH will not become alkaline, so it'll stay greenish in color. Any question about the citrate test? All right. The PR carbohydrate metabolism test, I think I've already talked a little bit about that. We're looking at the fermentation of a sugar. There will be one sugar in the tube. It can be glucose, lactose, sucrose, maltose, mannitol, or inulin. And I guess we don't have maltose in the table, but it could be any sugar you want. In the table, we only have the ones I mentioned, minus the maltose. And if the bacteria can ferment the sugar, it will make acid products. So it'll grow in the tube and appear cloudy. And if it can ferment the sugar, it'll make acid products, changing the color from pinkish or reddish to yellowish. And if it can ferment the sugar and produce a gas, we will get it turning yellow and see a gas bubble. And that's why in this test, this is referring to negative, that the sugar is not fermented. This is given an A, meaning the sugar is fermented, making an acid product. That's what the A stands for. And then this one is going to be an AG, which is acid and gas production. Any question about the carbohydrate fermentation test? All right. The motility medium is a semi-solid media that when you inoculate it with the bacteria, if the bacteria can swim, the bacteria can swim through this very thin agar. I think the agar is 0.4%. And normally the agar is around 1%. And it can't swim through usually 1%, but it can swim through 0.4%. And what you do is you inoculate the tube of solid agar. And if the bacteria are not motile, it will only grow in the stab. If the bacteria is very motile, the bacteria will swim everywhere in the tube, and you'll see the tube cloudy everywhere. Any questions about any of that? If the bacteria is slightly motile, it won't be everywhere, but it will grow beyond the stab, and we call it the bacteria flare out of the stab. So here's the stab. And let's make the agar this way. So the bacteria will grow something like that. A little more than just on the stab there, because it flares out. Let's make it a uneven one here, one there. something like that. So that's a flare. Any question about any of that? This bacteria is slightly motile. If it's highly motile, it would swim everywhere. And if it's slightly motile, you see it growing out of the stab. The stab mark, oops. If it's non-motile, it'll only grow on the stab. So it'd only be there. Any question about any of that? Motility happens in bacteria only if they have a flagella. If they do not have a flagella, the bacteria will be non-motile, because flagella is the only structure bacteria have for being mobile. All right, let's move on to the hydrogen sulfide test. This test is looking at the production of hydrogen sulfide. If hydrogen sulfide is made, then the tube will turn black. You can test for hydrogen sulfide production in the triple sugar iron or TSI test, in the sulfide indole motility or the SEMS test, and on hectone enteric agar or HE agar plate tests. All three of these can test for hydrogen sulfide production, and if hydrogen sulfide is made, then the area will be black. So here we have what is this? Must be the SEMS tube. And this one is hydrogen sulfide negative because it's not black. That one is very positive because almost everywhere is black. It is normal to see the darkening in anaerobic conditions, meaning below the surface. And we can see that here. And the surface is where the cells are not anaerobic. And when the cells are performing aerobic respiration, they do not make hydrogen sulfide. So they have to be engaging in anaerobe or fermentation before they make hydrogen sulfide. Anyways, that's one way you can test it. You can also test it with the triple sugar iron test. Let me just go down to the picture here. So this one is black. So we know that hydrogen sulfide was being made in this tube. And once again, we notice the black is in anaerobic conditions. The aerobic condition is not black. Any question about that? And then let's go on. There's SEMS again, where it's black when hydrogen sulfide is made. And then the HE plate, if the bacteria can produce hydrogen sulfide on an HE plate, the bacteria colonies will be black. Let me blow this up so you can see it better. So the colonies here are black, and that's because they can produce hydrogen sulfide gas. On this hand, this one here is not black, and that means this species cannot produce hydrogen sulfide, at least when growing on a petri dish. Any question about any of that? All right. The interpretation of the TSI test is complex because we have 3 or more tests running at the same time. You have fermentation of sugars, you have gas being produced, you have hydrogen sulfide being produced. Tube 1 is negative in everything. So K over K means alkaline over alkaline. You could also abbreviate that negative over negative. You go the aerobic, meaning the slant first for the tube. and then the lower portion of the tube for the anaerobic areas. So that's why K over K or negative over negative. No sugars are fermented. That's why the tube is orangish or reddish. No gas is being produced, and no hydrogen sulfide is being produced. No hydrogen sulfide because it's not black, and especially in the lower portion. No gas being produced because the auger is not broken or lifted. So here you can see there are cracks in the auger, and that's from gas being produced. There's a bubble in the auger, and there's a bubble at the bottom of the auger. And this is clearly showing gas being produced. Let's move on to tube 4. Tube 4 is yellow both in the slant and in the butt. So we know that lactose or sucrose is fermented, one or the other. The reason is because if only glucose were being fermented, it would be yellow in the butt, but the slant will turn reddish or pinkish. And I'll talk about that in a little bit. No hydrogen sulfide being produced because it's not black, tube 4. Let me talk about which sugar is being fermented. We know that lactose or sucrose is being fermented here because the slant is the same color as the butt. And in this test, the glucose is at 1/10 the concentration of the lactose or sucrose, meaning I think it's 1 gram is added of the sugar in the lactose or sucrose, and 1/10 of a gram of glucose is added. I think that's correct. If only the slant were to be red and the butt were to be yellow, we know that only glucose is being fermented, because if we had looked at this earlier, the slant would have been yellow. The reason being is that the sugar is fermented glucose, but when we look at it here, where the slant is pinkish or reddish and the butt is yellow, it's the butt is yellow because the glucose is being fermented, but the slant is growing slower. Excuse me, the butt is growing slower than the slant, and so the bacteria still have some glucose to ferment in the butt, and that's why the butt is red. However, if we had looked at the slant earlier, let's say 5 hours after you inoculate, the slant would be yellow, and that's because glucose is fermented, creating the acid products to turn it red. But the glucose gets used up because it's in low concentration. And that's just because in the TSI tube, the glucose is only 1/10 the concentration of the sucrose or the lactose. And so rather than die, the bacteria use the protein in the media, releasing ammonia as a waste product from digesting the protein or amino acids. And the ammonia is a base, and that will revert the yellow slant to a reddish or pinkish color. And that's why tube 3 is reddish on the top and yellowish on the bottom. And that's because only the sugar glucose is being fermented. Sucrose and lactose are not being fermented. This tube has no gas production. There's no breakage of the auger, like right there. No bubbles here or there. And it's not black, so no hydrogen peroxide was being generated. And then in tube 2, once again, we have a pinkish slant, and the butt is black. So obviously this one has hydrogen sulfide being produced. the auger is not broken up. There's no bubbles in there, or like there. And so there's no gas being produced in fermentation. But you know that fermentation is occurring because for black to happen, the tube has to be yellow, at least at the start. But the hydrogen sulfide will be hiding the yellow color. But hydrogen sulfide only happens, meaning is only produced in anaerobic areas, and that's why this is black. And if we'd looked at this tube earlier, you would have seen the extreme areas being yellow, and then this portion in the center black. So it's yellow here. You just can't see it. I just know it is because to be black, you have to be yellow. And it's only glucose that's being fermented because the butt would be yellow if the black wasn't there. And then the slant is not yellow. And so it's only glucose that is being fermented. Any questions about the TSI test? All right, let's talk about the sulfide indole motility test. It is a test that when you inoculate the tube, you can look at the production of hydrogen sulfide, you can look at the production of indole, and then you can look at the motility of the species inoculated into the SIMS tube. So you stab inoculate the tube, which I don't see it on the control there, but let me blow this up. On tube 3, we can clearly see the stab mark. On tube 4, we can see it, but it's not as clear as tube 2. And then tube 4, we have bubbling. The bacteria have not gone everywhere, but it has gone in this region here, especially the right side, and that's much greater than there or here. And so we can say that the bacteria on tube 4 is motile. The bacteria on tubes 2 and 3 is not motile. Tube 1 is, or 0 is also not motile. Tube 5 is not motile. Tube 1 is motile because the bacteria have gone everywhere, and that's why we're seeing black everywhere. Any question about the hydrogen sulfide production test in the SIMS tube? All right, that's the first test we run with the SIMS tube. The second test is whether the bacteria are motile or not. If the bacteria are not motile, they'll only grow in the slant where you stab inoculated. But if the bacteria are motile, they can either be everywhere like tube 1, or they can be mostly on the top right. I don't know why it's more on the right than the left, but it is. And then the third test ran from the Kovacs reagent, from the SIMS test, is the production of indole. If indole is produced, you then add the reagents, Kovacs reagent, and it will turn red if Kovacs is made, not Kovacs, whatever Kovacs reacts to, indole. Reddish or pinkish, and tube 1, tube 2, tube 3, and tube 4 have indole being produced. That's why it's reddish on the top, and only the top, by the way, because it's the red's going to be in the supernatant, which is not water. Which you can kind of see it there or there. And if indole is not produced, it'll be yellowish in color, tube 0 and tube 5. Any question about anything? All right, that's it for the indole test. I've already talked a little bit about the HE test. Let's talk about growth on McConkey's and HE. Both McConkey's and Hectone enteric agar are selective for gram negatives. So if you get growth on either the McConkey's or the HE plates, then you probably have a gram negative. These plates are selective because they have bile salts in them, which is something that's found in the intestine. And the McConkey's is also selective in that it has crystal violet in the media. And that's why it changes color to a purplish color. If you have the cells making a colony that can ferment the sugar lactose, they'll be pinkish in color, as well as the background will be pinkish in color. If the bacteria, on the other hand, cannot ferment the sugar lactose, non-lactose fermenting, then the bacteria will be its normal color. And that looks off-white to me, except for maybe there, which it's white. For the Hectone enteric agar, if the bacteria can produce hydrogen sulfide, they'll be black. If they cannot produce hydrogen sulfide, then they will not be black, and the colonies will be the correct color for this species. And this looks like it's white, but it could be off-white or some other color. All right, and then the second or third test on the HE plate is to look at the fermentation of the sugar lactose. On MAC, if the sugar lactose can be fermented, then it'll turn pinkish or purplish in color. Non-lactose fermenting, no color. And this is a gram negative, so it grows on both ends of the plates, which means it's probably an HE plate. For the let's go there. Oops, why did I get there? For the gram-negative species, which can grow on the HE plate, if they can ferment the sugar lactose, it'll change the color from yellow, orange, and this looks more like orange. I think I see some yellow in this region and that region there, in this region here. All right, any questions about growth on MAC and HE? All right, so everything you do in this lab will be done online, so we're not going to do any of these tests, but you will see videos on them, and you do need to understand the test, the biochemical test, and knowing how to perform and interpret these tests. So we have a video clip showing you carbohydrate fermentation. We have a video clip showing you sulfide indole motility test. We have a video on MRVP test. And remember, you inoculate this one tube with media, and you don't mix the media. And then you split the tube into half, one half doing the, what we did was like a piece of fruit. And then the other half does like the accumulation of protein in the cells, something like that. But the point is you inoculate with one tube, split it into two, and one half will do the MR test, the other half will do the VP test. You should understand about triple sugar iron. And I hate to tell you this, but maybe the easiest thing to do is just memorize this table, especially the concepts on the table, meaning it'll be black if you have the sugar lactose in there. If you have only lactose or sucrose or both metabolized, it will be yellowish in color, both in the black and the slant. If, on the other hand, only glucose is fermented and you are using the correct amount of time, which is 12 to 18 hours, I think it's actually 14 to 18 hours. And if the slant is pink, but the butt is yellow, it means that the bacteria used up all the glucose on the slant, and then rather than die, the bacteria started digesting the chairs. Any question about any of that? All right, we have a video on triple sugar iron. We have another video on McConkey's agar. Unfortunately, I don't have one on HE agar, but it's very similar to McConkey's. And then use the information in this lab module and previous lab modules to complete the worksheet for lab 9, which I got here. Case study 1. Go ahead and read about that. Case study 2. Oops, where is it? I'm trying to figure out where the worksheet is on this. I don't think I have it in there, which is a mistake. Let me just check your version. Come on. Close that one down. Oh, there it is. No, that's the module. We want the worksheet. Oh, there's the table. You need to fill in the table. I'm not sure why I don't have the table on mine. Maybe I'm looking at the wrong one. Yep, I was looking at the wrong one. That's why I didn't find the table. So for doing the lab, review how to interpret the following test by completing the chart, by adding a brief description. Oftentimes you can just put one word or 2 words. If you need to, you can expand the cell and add like a whole sentence or longer. But fill in the table as the first one has a result in it to tell you. For the oxidase test, the positive result is the cells or a spot turns blue, and then give me the negative result. Obviously, it's the opposite of that. And then for the indole test, that one is discussed in this lab. Give me when the positive result is and the negative result. For the MR test, it's also in this lab. Give me the positive result and the negative result. For the VP test, it's also in this lab. Give me the VP positive and the VP negative. And then for the positive result of the citrate test, you'll obviously have growth and it'll change color. You just need to say what color it is. And then the negative result. Tell me what the negative result is. For carbohydrate fermentation, we're looking at the sugar fermented, such as glucose. Tell me the positive result and the negative result. Now, for this one, you only need one answer. And then give me the gas being produced in carbohydrate fermentation, meaning if your inverted Durham tube of your test tube, which is, I believe, no, I guess we don't do anything in the lab, so it's not in the lab, at least for my lab. The gas produced, tell me what the positive result is. You'll see a bubble, and then the negative result. What do you see? And then go on and give me the TSI auger slant, which we discussed here. If glucose is fermented, tell me the result here. And if glucose was not fermented, give me the result there. And then if lactose and sucrose are fermented, give me the result there. And give me the result there. Now for these two, you need to put it with either negative over negative, and this can be negative, or it can be K over K. And that is, you put the slant first and then the but. Over here, it won't be negative over negative, so one of them would be maybe acid. And we're just going to make one up. And then K. Oop. Oh, I got that in the wrong place too. So for these two, you need to have two symbols, one for the slant and one for the but. Any question about any of that? All right. Give me the results of hydrogen sulfide being produced in the triple sugar iron. And then give me the negative, meaning hydrogen sulfide is not produced. Give me gas being produced in the positive and no gas being produced, meaning gas production negative. For the SIMS tube, we're looking at 3 tests: the production of sulfide, the production of indole, and the motility. For the sulfide production, give me the results here. For positive, give me the results for negative. And for the indole production, give me the positive indole being produced, and then give me the negative indole being produced. And then for motility, if the bacteria can swim, give me the positive answer and the negative answer. Realize with the positive answer, you may not get something growing really fast. And so you should say that bacteria are either growing everywhere in the tube or bacteria are flared from the stab. And give me the negative for motility. All right, and then the last table on the worksheet is selective and differential media. When the bacteria are grown on MacConkey's and HE, what happens with lactose fermentation? If it's positive, tell me what the I think I'm on the wrong one here.
Professor?
Yeah, sorry, I lost my
sorry.
place. You just finished the
Okay.
table. Yeah.
No, sorry. I realized the answer to what I was gonna ask.
Okay. With lactose fermentation, you should say the color that's positive for both MAC and HE because the colors differ. And then you should have the negative. And that's the color that's just normal. All right, then complete the questions on 2. For 2A, 1, what is the reagent that must be added to detect the presence of indole? So name the reagent. And if indole is present, the test will show what color. So give me the color there. For 2B, if methyl red is a pH indicator and it remains what color under acidic conditions, and then methyl red pH indicator is used in what test? Any question about any of that? All right. For C1, the what test uses Barrett's A and Barrett's B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism. If acetone is present, the test will be what color? D, we're looking at Simmons citrate. What's the pH indicator in Simmons citrate tube? And this pH indicator turns blue under either acidic or basic conditions. You can either bold or highlight or write it right there. And that's 2D2. And then for 2E1, what is the pH indicator used to detect the PR carbohydrate fermentation, like the PR glucose test? What's the pH indicator? Put it there. And then bold, write, or underline your answer here. And this pH indicator is yellow under either acidic or basic conditions. So give me the answer there. Or circle it or bold it here. Or you could write it there. Any questions about that? All right. F, bacteria that possess this structure test positive for motility. So what structure that bacteria have that gives them locomotion? So name the cell structure there. And then G, 3 organisms that produce hydrogen sulfide are 1, 2, 3, and there's at least 3 of them mentioned in the lab module. 2H, if an organism's ferment glucose, but not lactose or sucrose, the TSI slant will be what color? And the deep butt will be what color? Any questions about any of that? For I, state the 3 tests for which SEMS is used. For J, MAC and HE plates are what? In that they inhibit the growth of what? Meaning bold or right, gram-positive and gram-negative bacteria. So that's 2J2. And they are also what? In that they separate various bacteria based on visual differences from sugar fermentation. So that's saying it's more than just selective. So what are we saying here? All right, any questions about these questions? Any confusion? If not, work on the case studies and the worksheet. Let me go back to the case study here. I'll only do one, but come on, mouse, where'd you go? I lost my mouse. I don't know why. Oh, there it is. So MAC and HE plates are both what? And they inhibit the growth of what type of cell? Gram-positive or gram-negative? So give me what word inhibits the growth of the cells for J1. For J2, tell me which cells are inhibited, gram positive or gram negative. And you can bold this, highlight it, underline it, or write it there. And these plates are also wet in that they separate various bacteria based on visual differences from sugar fermentation. Any questions about any of these questions? All right. So in the case studies, go through the case studies and use this key to help identify the case studies. And this table should be filled in. For case study 1, tell me if it's oxidase positive or negative. Glucose can ferment glucose. Tell me it's positive or negative. And get as much in on this table as you can. Not all of them will be discussed. Like, I don't know if case study 1, they actually did the oxidase test. But let's go to case study 1 and case study 2. Am I in the worksheet? Yep. Let's go back to the lab module. Case study 2. Mikey talks his mom into buying a pet map turtle. And there it is there. Mikey does well at changing his water, but he's not necessarily diligent about washing his hands afterwards. About 2 weeks later, Mikey develops diarrhea, nausea, and fever. His pediatrician orders a fecal specimen for routine culture and a sensitivity test. So here we have the colonies grown on McConkey's plate, and the arrow is pointing at an important colony there. Oops. And here we have the bacteria growing on HE plate, and the arrow is pointing to a colony of interest. And then they do some other tests. They did the SIMS test here, the MR test here, the VP test here, the Simmons citrate test here, and then the triple sugar iron test here. So in case study 2, tell me, like, MR on the worksheet. I need to fill in case study 2. I don't know if they do oxidase test in case study 2. Yeah, they didn't do oxidase test in case study 2, so we can't answer it. However, it says that the bacteria are catalase positive and oxidase negative. So you do get the information. And the point is, come back to the lab module and fill in this table. So that one I'll tell you is positive. Oop. I'm not getting it. There we go. Took me a long time. And tell me if it ferments glucose, lactose, TSI, give the results of TSI, hydrogen sulfide being produced, whether it's modal, indole being produced. Let's see if there's indole in that. There is indole in the too, because we have Sims here and you add Kovacs reagent. And so there's the presence or absence of indole up there. All right, so fill in the chart there and then take a look at these tests and interpret what the tests are. So this is, I won't tell you what it is, but the MR test and the VP test. And then, oh, where do I have that? And then look at the case studies. So here's the case study here. And once you have this filled out, look and see what species it is. And then give me the name of the species under the case study here. Any question about any of that? All right. Go ahead and erase that now. So I'll be here until 8:30 or the last student logs out to help you with the lab. So go ahead and get started on the lab. Any questions before I stop the share? I won't stop the video. I'll be here to answer questions, but all right, no questions. Let's get started on the lab.
Hey, Dr. Volp, I had a quick question for you about the infectious disease project.
Okay, let me go ahead and start the recording just in case some. Am I sharing? No.
Have a good night.
All right, you too.
Thanks. Oh, and I don't think I need a meeting anymore, unless it's like me asking some questions that I can do on here.
I thought we already had the meeting. Did you
No,
want
we
to
did.
talk further?
Wait, did we?
Yeah,
Did
we had
we?
a meeting.
Oh, okay. Yeah.
Okay.
Okay, yeah, maybe we did.
Okay.
Okay. Thank you.
All right. Bye.
Okay, bye.
see. Oh, I'm the only one. End it.